Hello, Mr. Allman here, and this is your Allman Cast on genetic information. While we know today that DNA is the heritable information that's transferred from cell to cell and from parent to offspring, in the 1940s and 1950s it was still under vigorous debate as to how it was that genetic information was actually passed on from generation to generation and from cell to cell. Now, the work of two scientists, Alfred Hershey and Martha Chase, actually provided the proof that it was DNA and not, in fact, proteins that actually carried that genetic information. Now we start with, in their experiment, the T2 phage. A phage is a type of virus, and it is a virus that, like most viruses, is specific to a certain species. These phages have a particular shape that allows them to attach to and infect bacteria cells. It consists of a protein coat, so all of the parts on the diagram on the left in black, those are the protein parts of this particular virus. In the head, there is some genetic information there. And viruses at this time would have been able to have been isolated, and chemical analysis could have been done to be able to see that protein and DNA were the two components of a virus. Now, the way that a virus works is that it cannot reproduce on its own, but rather it uses a host cell to replicate more copies of itself. Now, because this is the case, if the proteins contain the genetic information, then the virus would need to inject proteins into the bacteria cell in order to show the bacteria how to be able to produce more viruses. Rather, if the virus inserted its DNA into the bacteria, then it must be the DNA that provides that information. So, in their experiment, they used these phages and utilized the chemical differences between DNA and proteins to be able to determine which one actually entered the cell. First, they started by radioactively labeling sulfur atoms that were incorporated into the proteins of the virus, because proteins contain sulfur and DNA does not. They then allowed these viruses to infect the bacteria and found that there was no radioactively labeled sulfur inside the bacteria cell. All of the sulfur that was labeled was actually still found in the protein coats of those viruses. When they replicated that experiment, but instead using radioactively labeled phosphorus, because phosphorus is an element that's found only in DNA and not in proteins, they found that the radioactively labeled atoms ended up entering into and were found inside the bacteria. This shows that it was DNA that moved from the virus into the cell and not vice versa. And so while prior to this experiment the majority of biologists thought that proteins, due to their variance and complex structure, were the genetic information, rather DNA is what is passed on from cell to cell. Now that brings us to a gene. A gene is a segment of DNA that encodes for a protein. Now there are many different segments of DNA that might combine together to be able to provide for the gene. If we use the example of beta globin is going to have a number of sequences to which regulatory proteins, things like transcription factors, will be able to bind. The part that's in blue, those are actually the sequences of nucleotides that are going to go through the process of transcription and translation and be used to produce amino acids. Therefore, there are whole large chunks of DNA that are never going to be used by the cell to be able to make proteins from them. That doesn't mean that those segments are not worth anything, and in fact we are still gaining our knowledge in terms of what it is that those sequences do. But those non-coding regions can be involved in things like turning on and turning off the transcription translation process. That's known as gene expression. They are segments that are cut out after transcription occurs. Those are called introns. And they can also encode for non-coding regions can code for things like tRNA molecules and ribosomal RNA, those things that have functions but don't actually specifically get turned into proteins. So even though the central dogma says that DNA is used to be able to make proteins, actually a lot of DNA are used for other things. Not all genes are used to build proteins or polypeptides, but the central dogma is a good place to be able to start in terms of your understanding of what DNA is used for. I also want to touch on mutations and different types. So DNA, any change in DNA can affect the protein structure, and proteins are the molecules that are doing most of the work in terms of the physiology of our body. So there are two major types of mutations that can occur. A mutation is a change, any change in DNA. If it's a point mutation, this is when we have one DNA base replaced for another DNA base. Another is when we have a frameshift mutation. So this could be DNA bases being added or taken away, called an insertion or a deletion. And this shifts the DNA triplets for the remainder of the gene, which can ultimately affect the way that that gene is going to be transcribed and translated. Now, sickle cell anemia is due to a type of point mutation called a substitution. The type of point mutation we see is that there was a T in the template strand of DNA. That is going to change to an A in the mutant form that people with sickle cell anemia have. That changes the mRNA to an A versus a U in those with sickle cell. And the translation of that, instead of glutamic acid, which is a negatively charged hydrophilic amino acid, we have valine, which is a hydrophobic amino acid. So that change in the translation ultimately affects the function of the protein. Now, not all point mutations can cause that change to occur. Silent mutations are ones in which the base change does not change the resulting amino acid sequence. So a change from an A to a G is going to cause a change in the mRNA, but because there are multiple mRNA codons for each amino acid type, it is possible to change a base without actually changing the amino acid that will be added. In other words, a different tRNA with a different anticodon will be brought to the ribosome during translation, but that different tRNA is actually carrying the same amino acid as the original sequence would have called for prior to that mutation. The majority of mutations are silent mutations because they occur in some place where the amino acid sequence doesn't alter. Again, a substitution is going to be the type that we see in sickle cell anemia. As we move over to frameshift mutations, these are often much, much worse in severity. A missense mutation is when a change in the base, in the example given here, is going to alter all of the amino acids downstream, or that are occurring after that mutation. So deleting 1A changes the phenylalanine glycine stop of those next three codons. That's changed to a sequence of 3 amino acids, leucine, alanine, and it continues on without the stop. A nonsense mutation is when the reading frame is altered so that a stop codon is added before the stop codon is supposed to be there. So as you can see in this case, we have methionine, lysine, phenylalanine, glycine, and then the stop. We have a sequence of amino acids with a stop at the end. However, an insertion or deletion can change the way that those codons are read, and putting in a stop before the stop is supposed to occur. So in this case, an extra base is added into the DNA, adding an extra base, a U, into the mRNA, causing the ribosome to read UAA, whereas before there was supposed to be a lysine, now there's a stop codon. The protein synthesis ends, and now we have an amino acids missing. We have a shortened polypeptide, and so now this protein may not fold, or it will fold incorrectly, leading to a non-functional protein. You can have a frameshift mutation that doesn't alter the codons, so you can see we have methionine, lysine, phenylalanine, glycine. If we take out a sequence of 3, and that sequence of 3 occurs at the original DNA triplet spots, then we can have methionine, phenylalanine, glycine. It's the same set of amino acids, missing the 3 bases that would have coded for the lysine. This can have some effect, but it's not necessarily going to cause the protein to stop functioning. So just to summarize the last couple things about mutations. Mutations change DNA, but not all mutations are equal. The majority of mutations are silent mutations that actually don't cause any change, either because they're in a non-coding region or because they don't actually change the amino acid sequence within a gene. It is only in some rare circumstances when the DNA is going to be changed and the resulting polypeptide will be changed. Most of those mutations are going to be harmful because they are going to alter severely the amino acid sequence, shortening, lengthening, or changing in a profound way the structure of that protein. So it is just a very small subset of mutations that actually change the amino acid sequence that through something like a substitution where the resulting damage to the cell is not lethal, but causes something like a genetic disease that we can then study, such as sickle cell anemia. On that point, sickle cell, if there was a mutation at any other point or a different type of mutation on the hemoglobin gene, we don't see that resulting in any particular genetic disease because it is a lethal mutation. Other changes are going to change hemoglobin in such a profound way that the cells would no longer be able to carry oxygen at all, and so no individual would be able to survive during the process of development with a mutation elsewhere on the hemoglobin gene. As always, if you have any questions, please let me know. Thank you.